So good morning, everybody. And this morning, we're going to carry on our discussion about biomolecules, these mostly very large molecules, which both construct the cell and which drive all of its metabolic processes. And last time we talked about two classes of biomolecule, and those were the carbohydrates, sugars, things like that. And then we also talked about the lipids, that's oils and fats in the body. Remember, carbohydrates are able to make polymers. They make polymers because they use monomers, which are simple sugars, and they join them together in various ways to form different substances and to perform different functions. Now today, we're going to talk about the next two classes of molecules, and these are things which you have definitely heard of before. First of all, proteins, and second of all, the nucleic acids, things like DNA. So these are things which are just nowadays so commonly referred to, and often we don't have a very clear understanding about exactly what they are. Well, proteins are immensely important in cells because they compose over 50% of the dry mass of the cell. If I dry a cell out, about 50% of it is protein. And the protein is in various forms. It's there performing different kinds of functions. Proteins perform a myriad of functions inside the cell, as we'll hear in a minute. We can look at the functioning of those proteins and class the functions as follows. First of all of our enzymatic processes, all the metabolic processes that go on in the cell are driven by enzymatic processes. So all of them have enzymes which govern the metabolic reactions. An enzyme is a catalyst. It's something that enables a reaction to take place very fast, where normally it would take place so slowly that it wouldn't be of any use to the cell. Instead, all of our metabolic processes are governed by enzymes which speed up those reactions and make them happen at an efficient rate. So there's a huge class of proteins which are enzymes. Most of our enzymes, 99.9% of our enzymes, are proteins. There's one exception, which we might mention later on. But for instance, proteins are used to build things, to break them down. They're used in both anabolic and catabolic processes. Remember, anabolic processes are building, catabolic processes are breaking down. And all the metabolic processes have their own enzymes. Enzymes are very specific to particular substrate and to particular reactions. And they speed up the rate of reactions enormously, make them sufficiently efficient for them to be useful biologically. So number one function are as enzymes. Number two, here's one that you've heard about frequently recently, because the proteins are also able to act in defense of the body. And they are one of the major products of the immune system in the form of antibody. Antibody is a protein, and antibody, its responsibility is to bind to things like viruses and bacteria, things like that, and inactivate them. So they are produced as a result of infection. It's a response of the immune system to produce antibody. The next use is as storage. This is most clearly seen in something like an egg, where a huge amount of nutrient is lodged in the form of protein, which can be accessed by an embryo. In an egg, for example, the white of the egg, that is almost entirely a substance called albumin, which is a very long, very big protein. And that's a store of nutrient for the embryo. The next function is, in actual fact, also a kind of enzymatic function, but it is a specialized function. And that is that proteins are able to govern transport of specific substances. So, for example, in the cell membrane, I told you the importance of the cell membrane was it allowed conditions to be different on one side to the other. That function is given to it by proteins which are embedded in the cell membrane. The cell membrane has proteins which select what substances go across in what direction. So they are responsible for transport. And most of the functions of the cell membrane are lodged in its proteins. It's the proteins that make the cell membrane a living thing that actually works to regulate substances going across. Very importantly, we'll hear about them again in some detail later on. Many of our hormones are proteins. And I'll just give you one example of this. The hormone insulin, which is released by the pancreas, is in fact a protein hormone, and it regulates the amount of blood sugar, the amount of sugar that's in your blood. It stimulates cells to behave to regulate the amount of sugar in your blood. But there are many other protein hormones as well. The next function is a little bit difficult to explain when we don't actually yet know anything about the way that nerves and things like that function. Proteins can act as receptors. You can think of them acting as a sort of a switch. Again, receptors are very often bound to the cell membrane, and they perceive substances in their environment, and they relay a signal to the cell. So, for example, here, this is a nerve ending, and this nerve ending needs to communicate, say, with a muscle, something on the side. And here, the proteins are embedded in the cell membrane. They are receptor proteins. The nerve cell releases substances called neurotransmitters, which bind to those proteins, and they cause the proteins to do something. The proteins now cause some process to take place. They don't transport the signaling molecule across. They just respond to its presence. They switch on, and they transmit a message to the cell. So those are receptor proteins. Receptor proteins are very important because if you think about it, you take a cell, just any cell. The cell has no eyes, it has no ears, and yet it needs to know all the time what is happening in its environment. So what conveys a lot of information to the cell about what is happening in the environment, what substances are present, what's the concentration of salt, what's et cetera, et cetera, et cetera. A lot of this information is conveyed by receptor proteins. They are the eyes and ears of the cell, and they're continually telling the cell what is going on outside. Next, if you just flex your arm, you are using contractile proteins in the muscles of your biceps. Those proteins are called contractile and motor proteins. They are proteins which are specifically dedicated to movement. And almost all movement inside the cell is governed by proteins. And this is a very, very, very complex thing, because we need, inside the cell, we need to try and be transporting stuff back and forth. We need to be done efficient. We need to send it to specific destinations. Second, that's just inside the cell. Second of all, we may be able to, maybe need to contract the cell. That move the cell, change its shape. That is governed by proteins. We may move one cell against another cell, as we do in muscles. And all of those movements are governed by contractile and motor proteins dedicated to movement. Dedicates to the movement of one sort or another. We'll talk about it a little bit. It's a complex topic. we'll talk about it briefly when we talk about muscles. The last is that much of the structure of a cell, and hence of the whole body, is defined by structural proteins. The cell itself has a skeleton, which is built of protein. And in addition, there are many protein is involved in many other ways in the structuring of the body. I'll give you one example. For example, the bones are not just non-living solid blocks of some substance. Calcium phosphate is what makes bones hard and rigid, but that calcium phosphate is embedded in a protein matrix. Bone is actually a living tissue, and its foundation is living protein, into which the calcium phosphate that gives bones its rigidity is embedded. So there's an example of structural protein. There are many structural proteins, but some that you may have heard of, things like collagen, and also a substance called keratin. And keratin is a protein that makes up your fingernails and your hair, and it toughens your skin. There's all sorts of responsibilities for keratin, and that is also a structural protein. So remember that what we have enzymatic, defensive, storage, transport, hormonal, receptor, contractile, and structural roles for proteins both in the cell and in the whole organism. So let's have a look at how proteins are actually made. Again, don't freak out when you see things like this. I don't expect you to remember all of the structure. I put it there for a reason, and I'll explain it in simple terms. Okay, so first of all, a protein, the most basic structure of a protein is that it is a polymer. And if it's a polymer, it consists of monomers which are joined together. In proteins, the monomer is called an amino acid. It has a specific chemical structure which makes it an amino acid. And there are only 20 amino acids. This is a remarkable thing, because you'll realize from what we've just discussed that proteins have to perform a huge range of functions in the cell. There are many different kinds of protein. They're all made of the same 20 amino acids. And they can be built just out of those 20 amino acids, because the amino acids, in fact, can be put together into any order. So that doesn't mean to say that they put together randomly. They're not. They're put together in a very strict order. And the order in which the amino acids are put together is determined by the genes. A gene is a recipe for a protein. It's actually a recipe for a polypeptide. And there's a little distinction between polypeptide and protein. All proteins are, in fact, polypeptides. But a polypeptide is not yet a protein. A protein is a mature molecule. It is a mature functional molecule. The polypeptide is simply the string, the original string of amino acids put together. That is the polypeptide basis of the protein. Various things happen to take a polypeptide and turn it into a mature functioning protein. So the polypeptide is a polymer, and it's a polymer of amino acids. The proteins vary in length. There are some which are short, but many of the proteins are extremely long. More than 1,000 amino acids joined together is not uncommon. Certainly, many are hundreds, but many are also thousands of amino acids long. So I just want to show you what an amino acid looks like because we need to explain how it is that these monomers can be joined. Any monomer can be joined to any other monomer. Any amino acid can be joined to any other amino acid. What makes them an amino acid, just briefly, is that they have a group here called the amino group. They have a group here called a carboxyl group. This is what makes them acid here, and this is what makes them amino. So the amino acid. Every amino acid has this. In all 20, that is identical. They all have that structure. And that is what allows us to join any amino acid to any other amino acid. The character of the amino acid, what it does, is determined by another group. It's a side group, and it does not participate in the formation of the polypeptide. So the amino acids differ from one another enormously in their character. Some are soluble, some are insoluble, some are polar, some are non-polar. It's all sorts of different characters to the amino acids. So again, don't worry about I'm not expecting you to remember. I just want to show you this, to show you how very different the amino acids can be. This is only 9 of the 20 amino acids. They differ from one another enormously because of the nature of these side chains, right? These ones here, this here, I'll just point out to you, look at these and look at them. Look at those side chains. They look like hydrocarbon chains, right? They are hydrocarbon chains. Do you think those side chains will dissolve into water very easily? No, they're not. They behave like hydrocarbon. They behave like lipids. These will behave very strangely with water. They will repel water on this by these groups. They are hydrophobic amino acids. Whereas these ones, this one here is highly soluble. Okay, so this is just you don't need to remember these side groups. This is just to point out to you how very, very different each amino acid is from each other. Now, I just want to point out one thing. If I was to make a protein and I was to put string together, I could string together 50 of these amino acids, leucine. I could string together 50 leucines, or 50 leucines and isoleucines mixed up together in a long string. That whole area of protein would be hydrophobic. It would be repelled by water. If I had a long protein molecule and I had a length of it which was all leucine, isoleucine, hydrophobic, it would be forced away from the water, and it would could cause the protein to begin folding. It would be pushed into the center of the protein molecule. This is how these groups begin to influence the shape and everything of the protein. And it's these side groups, it's the way in which they're grouped, it's their sequence that determines the character of the final functioning protein. So they differ by side groups, very, very different kinds of behaviors, very different chemical behaviors. But look here, they each have that group there, 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 there. And we're going to use those groups to join them together into the polypeptide, because each of them has an identical root which can be used to form the chain. And it's formed by taking out a water molecule and forming what is called a peptide bond. Here are two which are already joined together. There's the bond there. That's the peptide bond, and it's joined this one to this one. Now we want to bring in another one, another amino acid, and join it onto that chain. Here it comes here. And enzymatically, using other proteins as enzymes, we will take out a water molecule and form a new peptide bond. All you need to remember, first of all, this is a dehydration reaction. We're taking out water to form a polymer. Second of all, in proteins, the bond is a peptide bond between amino acids, okay, between amino acids. The rest of the chemistry, you don't need to know. So proteins, these very big molecules usually, and their first of all, the initial impact on their function is the sequence of amino acids. That's called the primary function. It's just a sequence of amino acids in that polypeptide. But the different amino acids begin to interact with one another in the chain, and different areas of that chain begin to interact with one another as well, and they force the whole thing into a complex three-dimensional shape in space. And that three-dimensional shape is what determines the function, because it puts to the outside, or it creates areas with specific chemical character in the protein, and that's what gives the protein its function. So this here is an enzyme called lysozyme, and lysozyme you actually have in your tears of your eyes. It also is present in the nose because it originates in the eyes. And lysozyme is an enzyme which actually breaks down the cell walls of bacteria. So it's an antibacterial substance in your tears. It's very important. It's one of the things that protects your eyes from infection. And here it is. So these are just here to show you. This is a way of representing just the basic structure, and you'll see it can be very complex. There's all sorts of helices and bends and everything else. All of this structure is determined by the reaction of one amino acid with others in its immediate vicinity, and then the reaction of areas of that polypeptide to other areas, pulling it into structures like the helix, and then moving whole areas of the molecule together. And we end up with this very complex three-dimensional shape in space. And I'll just tell you that for lysozyme, this is the key area here. This is where this groove here is where all the activity of the enzyme takes place. That's where it is able to bind to bacteria and dissolve them. And that we call the active site. So this folds to move particular amino acids with particular character into a specific area with specific function. And that is the active site. So just to show you how very important that primary structure is, I'm going to show you what happens when a mutation occurs in a gene. Now, I remind you, did tell you before that by and large, DNA is replicated and is passed on from one cell to another, from one organism to another. It's usually passed on very, very accurately, but every now and again, a mistake may arise. Sometimes it doesn't make any difference. Often it is very bad. When that mistake is introduced into the DNA, it is called a mutation. And many mutations are, in fact, deleterious. Here is an example. And this produces the disease called sickle-cell anemia, sickle-cell disease. It's an inherited blood disorder, predominantly in people of African American descent, and very largely in people whose ethnic origins are in West Africa. Although it does occur across Central Africa, it's predominantly West Africa. And that's because it's been inherited back and forth within a prescribed geographic area. And sickle cell disease can be a devastating disease, and it results from a change in one single amino acid in the protein hemoglobin. So now again, don't freak out. We're not going to worry about most of this. But hemoglobin is the protein which is present in red blood cells, and its responsibility is to carry oxygen So it carries oxygen to tissues which are depleted of oxygen. It releases the oxygen and it returns to the lungs in the blood corpuscles to be reoxygenated. That's its normal function. So here's its normal sequence. And just keep your eyes on this here, on the number amino acid 6, which is glutamine. And in the normal circumstances, there somewhere way back in human evolution, a mistake was a mutation entered into the human genome, and people began to inherit this particular change. And it was a change in the DNA which changed one single amino acid. And here it is here. We have glutamine changed here to valine. And that produces a very profound change in the function of hemoglobin. Normally, hemoglobin kind of, I don't know how to describe it, kind of expands and contracts to when it picks up oxygen and then it releases. And it goes back and forth. It returns always to an original shape and then back. When you introduce this single amino acid change, when the hemoglobin releases its oxygen in the tissues, instead of returning to a normal shape, it forms dense fibrils like this. It actually kind of crystallizes out inside the red blood corpuscle, and it collapses the blood corpuscle into this distinctive shape. That's where it gets the name sickle cell anemia. These sickle cells are very poor at carrying oxygen, and in addition, they're very bad at passing through capillaries and the very fine spaces that they need to in the periphery and in the organs. And so they tend to start clogging up the blood vessels and causing all sorts of dangers. And sickle cell anemia can be a devastating disease. It varies in its severity. Some people have it and manage perfectly well. Other people are devastated by it and can actually die of sickle cell disease. The hopeful thing, sickle cell disease is one of the diseases which within 20 years or so, I would predict we will be able to cure through genetic manipulation. We'll be able to restore people's genes to the normal sequence instead of them having to carry this. So again, I told you, the first thing that happens, we got the primary sequence, and then all of those amino acids begin to interact with amino acids in their immediate vicinity to cause three-dimensional structure to rise, and then whole areas of the protein chain begin to interact with one another to fold everything into this three-dimensional structure. And the three-dimensional structure is critical. That's why hemoglobin doesn't work in sickle cell, is because its shape has changed. It has been changed by that single amino acid change. But there's something else about it, protein structure, and that is that a lot of these interactions are affected by environmental conditions. And so, for example, changes in pH, in the acidity or alkalinity balance, changes in salt concentration, changes in temperature, all sorts of things can cause the shape of the protein to change. And very often we'll find that the proteins have an optimal pH, an optimal salt concentration, an optimal temperature at which they work best. And if we change those conditions, they stop working well, or else we can change the conditions so radically that the protein actually kind of unravels, or other drastic things happen to the shape of the protein, and it no longer functions. And that we call denaturing. So a denatured protein stops being able to function as it normally does. Under certain circumstances, they may be able to return if we restore the conditions, but often they can't. The protein is effectively killed if you drastically alter its conditions. a simple example of this, albumin in the white of an egg. That is a protein. It's normally gel-like. It's very, very long. So it's liquid, but it's kind of a jelly. That's its normal function. Heat that up, and you cause it to permanently bind one molecule to the other tightly. And that is what happens when you cook the egg. The white of the egg is denatured albumin, and you can't restore it to its normal function by cooling the egg. It just stays solid. It's permanently denatured. Okay, so that's all that we're going to talk about with proteins for the moment. We'll hear reference to proteins again when we talk about things like muscles, enzymes, etc. But I just want to very briefly also introduce you to the hereditary materials of the cell, which we hear referred to so often, and yet we often have very little understanding of what they are. DNA is a familiar term to you. You may not have heard of RNA, things like this. These are all nucleic acids. The importance of DNA in the cell is that it is the DNA that determines the sequence. DNA contains a recipe for this sequence of every protein. Now, you've learned already how important the proteins are in the cell. Not only make up most of the cell, but they drive all of its processes, etc. Each protein has its own gene in the DNA, which determines the sequence of the DNA, of the amino acids, rather. So you can think of it that DNA is organized, and it's organized into units called the gene, and that is a unit of inheritance. And the DNA gene is contains a recipe for a protein. It contains the recipe for that protein sequence. So DNA is a nucleic acid. There are many nucleic acids in the cell. DNA is one of them. There is a problem for DNA, though. It can't speak directly to the cell. You can't read off a protein directly from DNA. Instead, there's a complex cell process to produce the protein, and the DNA has to be transcribed into another nucleic acid in order to talk to the cell. So it doesn't talk directly, it talks indirectly to the cell. And the nucleic acid that it talks to the cell with is called RNA. And I'll show you the difference in a minute. The DNA, we go DNA, it's transcribed to RNA, and the RNA is translated then into protein. And that is a process, by the way, called the central dogma. DNA is transcribed to RNA, which is translated to protein. And it's called the central dogma because that is the way every living thing works. Every single living thing, every living process is determined by that sequence of events. DNA is transcribed to RNA, which is translated to protein. So we have two kinds of nucleic acids in the cell that we're going to talk about. The first is deoxyribonucleic acid, DNA. The second is the nucleic acids which function inside the cytoplasm, and those are the RNAs. There are several of them, but it's ribonucleic acid. Ribonucleic acid. DNA, the original recipe. RNA, it has many functions, but the one that we're going to understand first is that it's a messenger. There's messenger RNA, which takes the recipe of DNA and allows the cell to see what it is, translate it. So DNA is really important because it's got all the recipes for the cell, but we need to think. Whenever a cell divides, it's got to hand those recipes on to all of the resulting cells. When an organism reproduces, it's got to hand on complete recipes for all of the functioning of the organism to all its progeny. So DNA, we have to be able to double up DNA continuously. We have to be able to replicate it so that we can replicate the recipes, hand them on to new cells as new cells are produced. And DNA does this very easily. It's quite a simple process. It's complex because it's a biological process. All processes in biology are complex. But at its basis, it is a simple process, and it's a simple process to understand. DNA and RNA are not actually that complicated. And I'll point out immediately that all of the nucleic acids, I've told you before, all of the nucleic acids are composed, they are polymers, and they are composed of just 5 monomers, which are called the nucleotides. So there are only 5 nucleotides, and yet we can build these incredibly complex molecules, DNA and RNA, out of them. And again, it is because we can join any nucleotide to any other nucleotide. So that's not to say they join together randomly. Of course they're not. Their sequence is critically important. The sequence of nucleotides is what determines the character of the gene. The character of the gene is what determines the protein that it's going to produce. But DNA is easily replicated inside the cell. In fact, the DNA locked into the structure of DNA is the recipe for its replication, as I'll show you in a minute. So that process, DNA is transcribed to RNA. That process produces a form of RNA called messenger RNA. And it's literally that. DNA is transcribed to messenger RNA because messenger RNA goes out to the cell to be read and to have protein produced from it. So messenger RNA goes to the cell, and there is a special little unit inside all cells called the ribosome, which is able to read or translate messenger RNA, and it produces the protein. It is the ribosome that produces the protein, and it produces it using the messenger RNA as the recipe. So that is how protein synthesis is controlled. There's the original recipes in the DNA. It's read off as messenger RNA, and the messenger RNA is then translated by the ribosomes. The messenger RNA is translated by ribosomes to produce the protein. We won't discuss the whole process really in any more detail than that. Again, don't freak out at this whole diagram, but I'm just going to use it just to show you very briefly some of the features of nucleic acids in general. First of all, this is a nucleotide, and this is the monomer. This is the monomer of nucleic acids, and it consists of a sugar molecule here, and attached to that sugar molecule there is a phosphate group there. There's a phosphorus atom there, and it's got lots of oxygens, so that's a phosphate group, right? Here's the sugar, and very, very importantly, attached to that sugar in each nucleic acid there is one of 4 nucleotides. So there's one of 4 I'm sorry, not nucleotides, nitrogenous bases. Excuse me. Attached to each sugar is one of 4 different nitrogenous bases. And in DNA, those 4 are cytosine, thymine, adenine, or guanine. Okay. Cytosine, thymine, adenine, or guanine. C, T, A, or G in DNA. So you can have there are 4 different nucleotides, in other words. They all have this as their basis, but they differ here. Where have you heard that before? Monomers very frequently have a part which is identical. And it won't surprise you to learn that when we build a nucleic acid, the nucleic acids can be thousands upon thousands of these units of nucleotides, monomers, joined together. And here they are here. Here's just a single chain. And you'll see that what we've done is we're going to use this and this. We're going to join, there's the sugar, there's a phosphate, there's a sugar, there's a phosphate, there's a sugar, there's a phosphate, there's a sugar, there's a phosphate. So what we've done is we've made, in this molecule, we've made a long chain with this backbone, this sugar phosphate backbone. In DNA, it's important to note that that backbone, sugar phosphate, sugar phosphate, sugar phosphate, is very, very strong. DNA is inherently a very strong molecule, and its strength is given to it by that backbone. And it has to be strong because it's got to be incredibly long. It's got contained in the sequence here of nucleotides is the recipe for the amino acids that are going to make up a protein eventually. So it's containing a huge amount of information. They're often very, very big, and they are formed by this backbone. Now, sticking into the center here are the nitrogenous bases. These are just ring compounds which have got lots and lots of nitrogen in them. That's why they're called nitrogenous bases, because they're very slightly alkali, not very strongly, very, very weakly alkali. But they stick into the middle here. So again, we've got a chain, sugar phosphate, sugar phosphate, sugar phosphate. Each nucleotide has its own nitrogenous base, whatever it is, pointing into the middle. They can be a single ring, or they can be two rings. So I'll just tell you that the difference between RNA and DNA, there are two differences between RNA and DNA that you need to know about. And the first is that in DNA, it's the sugar here is called deoxyribose. It has no oxygen here. You see there's just an H, has no oxygen. So it's a deoxy. It had its oxygen taken away. Ribose has an oxygen there, OH. So in RNA, it's ribonucleic acid. It uses ribose. DNA, it uses deoxyribose as this sugar here. And it's a way of labeling these substances so the cell recognizes what they are. The cell recognizes DNA because it has no oxygen here. It recognizes RNA because it has an oxygen there. Don't worry much about the rest of it. Okay, the other difference that in RNA does not have thymine. It has uracil. So RNA would look much the same, excepting the four bases that it can use are cytosine, adenine, guanine, and uracil. That's the difference. It uses uracil instead of thymine. Okay, so we've got one strand here, but DNA is not a single strand. RNA is. RNA is a single strand of nucleic acid. DNA is not. DNA is a double strand. So how is that formed? Well, it forms the double strand by matching bases. It always has three rings across the center. And it always matches cytosine to guanine. It always matches adenine to thymine. And it doesn't matter what order they're in here. Okay, have a look here. Adenine, thymine. This is DNA, right? It has thymine, not uracil. Adenine, thymine. Cytosine, guanine. Here's a guanine. Guanine, cytosine. Thymine here, adenine. And it matches them across there. And those bases, those matching bases, they link up with one another with very weak hydrogen bonds. So there are two sides to a DNA molecule. There's a sugar phosphate backbone down the outside. Then across the center, there are these nitrogenous bases, always three of them across the span: adenine to thymine, cytosine to guanine. and those are linked with weak hydrogen bonds. So this is very, very important. This is very, very important because now we look at DNA and we recognize two things about it. First of all, locked into this sequence of nucleotides here is the recipe, and that is the gene. In fact, their three bases together form what is called a triplet, and each triplet codes for one amino acid. So we can use this sequence here. We use that sequence as a recipe for the proteins produced by the cell. The second thing is, just imagine if I now come along and I separate this. If I separate one strand from the other completely, okay. Let's in our imaginations, let's separate these two strands and say I've got one strand here which reads CACG, and I don't know what the strand should be. I can create the strand quite easily, because if I have got one complete strand here that says CACG, I know this strand must read GTGC. Similarly, if I have GTGC and I've destroyed the strand, I can recreate it GCAG because I know what matches here. That's what we mean when we say the DNA contains within it the recipe for its own replication, because all we've got to do is separate those two strands, one from the other, and recreate the strand down the other side. And that's exactly what DNA does when it needs to replicate. When a cell divides, takes all of its DNA and it splits it apart and it recreates. It doubles up the DNA by recreating the matching strand. I'll just very simply tell you, usually, RNA is single-stranded, but RNA has many, many functions inside the cell, and a lot of its function is tied to its structure. RNA, although it's single-stranded, it can match up different areas of the same molecule, one to the other, and start to form these kind of complex shapes. This is a single strand, and I'll show you how it's a single strand. It's like a roller coaster. You know, we would go. We go here, run down here. Whoa, 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 and off here. And that's a single strand. Okay, but it's linked up. Some bases have linked up with one another using the same matching rules, excepting, remember, RNA has uracil, it doesn't have thymine, still matches to adenine, AU instead of AT. In DNA, A to T, in RNA, A to U. But RNA is a single strand, but it can be formed into very, very complex structures by this kind of bonding. Okay, so that's it for that section, and I'll see you again on Friday. And I just remind you that these classes now, I am posting them as MP4s because some of you still continue to have difficulty accessing through Zoom. So now we'll do it with MP4s to see how well that works, okay?